You can look at a mango resting on a kitchen counter and then think about the invisible bacteria clinging to your hand, and it's almost impossible to perceive any shared physical traits. Life on Earth looks incredibly diverse. Look at a blue whale. It spans up to 100 feet, weighs as much as 30 elephants, and navigates oceans through sheer biological force. Yet beneath that massive scale, the whale, the mango, and the bacteria are constructed from the exact same basic unit. Every living thing that has ever existed is built from microscopic machines called cells. But if you look closely at how those cells are built, a sharp dividing line appears. Every living organism belongs to one of two categories, determined entirely by an internal architecture designed billions of years ago. Tracing the line between these blueprints reveals how life scaled up from invisible specks to the enormous, complex beings we see today. The older, original design is the prokaryotic cell. This is life stripped down to its absolute essentials: ancient, entirely single-celled, and highly efficient. Because they rely on this simple design, prokaryotes are incredibly tiny, measuring anywhere from just 1/10 of a micrometer to 5 micrometers across. Inside, a prokaryote operates on an open floor plan. There are no internal walls or membrane-bound compartments. All of its genetic instructions, usually a single circular strand of DNA, just float freely in a central area called the nucleoid, completely unshielded. Today, this stripped-down architecture is used exclusively by organisms in two domains: bacteria and the extremophiles known as archaea. Running such a lean operation makes these organisms brilliant at surviving and reproducing quickly. But without internal organization, they hit a physical wall, permanently restricting them to a microscopic scale. To break past that wall, life required a massive architectural upgrade. That upgrade is the eukaryotic cell, built specifically to handle highly complex systems. Instead of an open floor plan, eukaryotes use internal membranes to compartmentalize their space. They build dedicated rooms, called organelles, to isolate and optimize specific chemical reactions. The defining feature of this new layout is a heavily guarded central vault. This is the nucleus. Instead of a single floating circle of genetic code, eukaryotes store multiple linear chromosomes safe inside this membrane-bound room. Every plant, animal, fungus, and protist on Earth runs on this advanced architecture. By enclosing specific tasks in closed rooms, the cell can safely manage acidic environments for digestion or high-energy power generation without destroying its own internal machinery. When you place them side by side, the physical gap between the two becomes obvious. A typical eukaryotic cell is 10 to 100 times larger than a prokaryotic cell. You are comparing a single-room studio apartment to a sprawling mansion. The mansion is packed with specialized machinery. Organelles like mitochondria generate power, while the endoplasmic reticulum folds and transports proteins. The studio apartment has none of these. The prokaryote keeps its single circular DNA out in the open, while the eukaryote locks its linear DNA safely behind the walls of the nucleus. To multiply, the prokaryote simply copies its DNA and splits in half. The eukaryote, carrying far more genetic baggage, requires the highly orchestrated steps of mitosis or meiosis to divide. Yet both models are undeniably cells. They each retain a protective plasma membrane acting as a selective border, a gel-like cytoplasm filling the interior, and tiny ribosomes to build proteins. The eukaryotic design isn't necessarily superior at basic survival. Bacteria are perfectly adapted for what they do, but that internal organization is the strict prerequisite for building anything larger than a single isolated cell. This architectural divergence dictates the ultimate physical ceiling of an organism's potential. The unpartitioned design of the prokaryote guarantees it will always operate as a lone survivor, trapped in a microscopic existence. By evolving internal organization, the eukaryotic cell solved the problem of scale. The protected environment allowed individual cells to specialize, adapt, and physically link together. Once cells could cooperate and divide labor, they could form tissues. Tissues could form organs, and organs could build the highly complex bodies of animals, plants, and humans. Every leap in biological scale traces back to the moment a cell built a wall around its DNA.